All right, good morning, everyone. My name is Robin Wolf Scheffler. I'm from the MIT program on Science, Technology, and Society, and it's my pleasure to introduce this panel on revolutions. As you've seen this morning, people commonly talk about the life sciences in the modern era as one of revolutions: the revolution of DNA, the revolution of informatics, the revolution of gene editing. As a historian of the life sciences, however, I'm not quite sure if this is the metaphor we want for talking about the work we do here at MIT. A revolution, if you'll remember, implies a violent turning over of the world. Indeed, philosophers of science who have looked at revolutions have gone so far as to suggest that the worlds of science before and after a revolution are incommensurable with each other. There can't be any dialogue, only misunderstanding. Speaking in revolutionary terms, therefore, risks obscuring the rich history of work at a place like MIT and dismissing its importance to the present day. And I think this gives us a shallow understanding of the types of innovation and cooperation that make our institute unique. A better metaphor than revolutions, if I'll be forgiven, is that of waves. If you think about a stream of drops landing on a pond, and the force of each wave might dissipate. The discovery of DNA becomes ubiquitous as a tool for research over time. However, it's still there. It still shapes every subsequent type of work. New drops in the pond add and interact with others that came before it, creating new and unexpected patterns. And I think this sounds much more like the work that's happened at MIT. As we've heard from Professor Sharp, and we will hear from the speakers in this panel, ideas and institutions build upon each other rather than violently replacing or superseding one another. New people and new ideas find new uses for old tools, and they combine them with their new knowledge. The story is not one of displacement, but of constructive interference and intermingling of science and engineering to produce unexpected and new ways of knowing life that could only happen here. MIT is so special precisely because our pond, if you will, has seen so many drops land upon it. So, without further ado, it's my pleasure to introduce three speakers who have both made their share of waves at MIT and have seen many wash upon its shores. First, Tyler Jacks, who is the David H. Koch Professor of Biology, a Daniel K. Ludwig Scholar, the co-director of the Ludwig Center at MIT, and he's going to speak to the long history of assembling genetics, informatics, and technology to study cancer. Second, Douglas Laufenberger, who's the Ford Professor of Engineering and a founding professor of the modern Department of Biological Engineering. He's going to speak to the entry of engineering approaches into the study of life as a novel way of studying and doing science. And finally, Ruth Lemon is the director of the Whitehead Institute and a professor of biology. She is going to highlight the long work of the Institute in combining approaches old and new to understand the complicated and rewarding question of life's complexity itself. So without further ado, I'd like to bring Tyler Jacks to the stage.
Well, thanks, Robin, and welcome, everybody. Thanks for joining us. I'm personally extremely excited that we're here today celebrating the launch of MIT HEALS. As you heard, I was involved with Chris Prather in leading a faculty committee that led to a series of recommendations that arguably brought us to this point, and I'm just so excited about what this collaborative will do for our community, for our region, and really for the world as it relates to advances in life science and, importantly, in health. I've been asked to talk a little bit about cancer research at MIT and tell the MIT cancer research story, which I'm very happy to do. It's a long story. It goes back, as you heard from Phil Sharp, 50 years now. In fact, we're celebrating the 50th anniversary of cancer research on the MIT campus this year. It began sort of modestly in this converted candy factory on Ames Street, where MIT decided to invest in molecular biology and recombinant DNA technology to understand the mysteries of cancer. And that effort, as you heard from Phil, was massively successful and really laid the foundations for modern-day molecular oncology. Over the years, many MIT faculty got interested in cancer research. It really touches every corner of our university. And in response to that, we've created the Koch Institute, which embodies this spirit of collaboration, as Phil called it, convergence, bringing together multiple disciplines to address and hopefully overcome the challenges of cancer. The Koch Institute is a physical entity, but it also connects many aspects of the MIT community around these most important problems. And I'll touch on some of the activities that the Koch Institute faculty are working on today and sort of project on into the future. But I want to begin looking backwards again to the Cancer Center. This is a picture similar to one that Phil showed you of a group of investigators who were recruited to the Cancer Center, including Bob Weinberg, Nancy Hopkins, who was showed in the previous image, David Baltimore, Salvador Luria, Phil Sharp himself, thinking about how do cancer cells arise from normal cells? What are the changes at the molecular level that drive that process? And Bob Weinberg, who's circled here, was interested in the question of whether normal genes are altered through mutation in the development of cancer cells from normal cells. And he set about to find the first cancer gene. And after some years of effort, he and his lab group published 3 really landmark papers in 1982 describing the first human cancer gene. It was called HRAS, which is mutated in bladder cancer. That was where it was found. But it and its cousins are now known to be mutated in many different, very important human cancers, including lung cancer and colon cancer and pancreas cancer and more. It really is an important driver of the cancer process. It becomes an oncogene through a very subtle change in the DNA, a single nucleotide change, which leads to a single amino acid change, and that converts the protein to an oncogenic form. And the hope of that research was that if we could understand cancer at this level, we could actually address it molecularly and maybe find new drugs that target this altered protein and similar altered proteins and bring about more effective treatments for the disease. That has happened, and I'll touch on that briefly. It happened here too, but it took a long time. RAS turned out to be a very difficult problem, and it actually took nearly 40 years. Investigators from academia and then industry to target altered RAS. But in 2001, first Amgen and then Mirati, now owned by BMS, successfully developed KRAS targeting drugs, which have been approved by the FDA in lung cancer and more recently in colon cancer. And this represents a major shift in our ability to control those cancers that I mentioned that have KRAS and other RAS gene mutations as drivers. So that was then a single cancer gene, 1982. This is now where, with the advent of high-throughput DNA sequencing technologies, the sequencing of the human genome, as you heard from Phil, major contributions by the Broad Institute and others at MIT, we now have collectively sequenced probably close to 1 million cancer genomes by now, and discovered not just one human cancer gene, but probably 500 cancer genes that are altered by mutation in human cancers. And there's more than 100 molecularly targeted drugs like these that I'm showing you here that have been approved by the FDA for the treatment of cancers with those alterations. So the promise from 1982 is actually borne true in spectacular fashion. These genes are mutated sometimes rarely in a given cancer, but sometimes commonly across many cancers. This is an analysis done by investigators in a large international collaboration, including the Broad Institute investigators, looking at now 26 commonly mutated cancer genes across here, 26 different cancer types. And again, this allows for molecular diagnostics to identify particular cancers with particular alterations that allows them to be treated with targeted, more precise cancer medicines that are having significant benefit. Now at MIT, we're approaching the complexity of cancer, this myriad of mutations in cancer genes, in lots of different ways. We have expertise in my lab and elsewhere at the Institute in modeling the molecular alterations in cancer in genetically engineered mouse models. And we can do this using a variety of techniques to very accurately model what happens in human cancer cells, but in a tractable experimental model system, the mouse. And these tools are useful not just to understand the molecular development of the disease, but also what one can do about it. How can you use these models to detect cancer at earlier stages, or treat it more effectively, or maybe intercept its progression from its early stages to its later stages? And this is very active and fertile ground here at MIT. The technologies for making genetically engineered mouse models have improved dramatically over the years. I started this work myself here at MIT in 1988, and it was very difficult to do back then. Now with tools like CRISPR, we can do this very, very easily. And the most recent version of CRISPR tools that we use in this process is called prime editing. method developed by David Liu at the Broad Institute, which takes the Cas9 enzyme, modifies it so that it no longer makes a double-stranded DNA, but rather a single-strand Nick, and then fuse that nickase to a piece of reverse transcriptase, an enzyme that will take an RNA template and convert it into a DNA form. This was actually a discovery made by David Baltimore, which was the thing that won him the Nobel Prize in 1975. And you can therefore feed this enzyme a long extended guide RNA that will bring it to a place in the genome, but also have an extension on it that has a mutation shown in red here. And the reverse transcriptase activity will then convert that mutation in RNA form into DNA, and that DNA will then be transferred into the genome at that specific position. This is an incredibly powerful technology. We've used it in collaboration with David's lab to make mice that carry the prime editor. And now in a simple system where one designs a guide RNA carrying a mutation of interest, one can introduce that into animals and mutate genes at any position in vivo. And we've done that in one example, and bring us back to the RAS oncogene, to look at different alleles of KRAS and how they affect tumor development here in the lung. And I won't belabor this point, but suffice it to say that different mutations cause different phenotypes. They all cause cancer, yes, but some of them, G12R here, causes a much more profound cancer phenotype than the other three, for reasons that we don't understand. But in all likelihood, this is teaching us something very important about the biology of RAS and how ultimately we will want to target it. We can do this in a gene by gene and allele by allele fashion today, but we are also able to use massively parallel methods using CRISPR-based libraries to mutate many genes simultaneously or create many alleles of genes simultaneously. This is the work of Francisco Sanchez Rivera, assistant professor in the Koch Institute, who's developed methods to introduce via prime editing whole libraries of guide RNAs to mutate a given gene of interest at every position to create a massive number of alleles across that gene, ones that are known to be mutated in cancer and others that perhaps are not yet known, but might be discovered in the future. And by this method, by introducing a range of mutations in a pool of cancer cells, one can then subject them to a variety of functional tests to determine the consequences of these individual alterations, and again, help predict what will happen when we find these mutations in the clinic in the future. Now, I've spent most of my time this morning talking about biology and molecular biology and genetic engineering, but as I mentioned, the Koch Institute is also about the convergence of engineering and science. And so I want to give a few examples of technologies from the engineering domain that are actively being pursued and are impacting how cancer patients are treated and also how we discover cancer in the future. Phil mentioned that one of the focuses of our research is to find new ways to deliver medicines to cells, including cancer cells. And we have great expertise in nanotechnology in Dan Anderson's lab, Paul Hammond's lab, Bob Langer's lab, that are leaders in this space. But we're also interested in using new engineering-based methods to deliver more conventional drugs, including conventional chemotherapy. You might be surprised to learn that chemotherapy is used today, and the dose of chemotherapy is used today based on formulas that were developed more than 100 years ago that simply ask the body size of the individual who's being treated based on their height and their weight. And by this very simple formula, regardless of the particular individual, whether they be very tall, short, and heavy, they might get the exact same dose of drug. And the distribution of the drug in those two different people is likely to be very, very different. And their genetics is likely to be very different. And their metabolism is likely to be very different. But the formula doesn't pay attention to any of that. And so Louis de Ritter, a graduate student in Bob Langer's lab and Gio Traverso's lab, working with collaborators at the Dana-Farber, decided to try to bring technology to solve this problem in a very simple fashion, to develop a closed-loop system that would actively measure the concentration of a drug, like a chemotherapeutic, while the patient is being treated, to test the concentration of that drug in real time, and then adjust the dose as a function of the measurement of the drug in the bloodstream. They call this system Claudia, and it's not just an idea, they've actually built it. Again, the concept with Claudia dosing is that one can actively measure and therefore monitor and adjust dosing so that the dose of the drug in that individual patient will be at the desired amount. Whereas with the body surface area dosing, shown on the lower left, one would overshoot in some cases, undershoot in others, depending on the various factors that I mentioned previously. They've built this device, they've tested in an animal model so far, and what you can see from these data is that compared to the standard approach, which overshoots in this particular example, leading potentially to overdosing, with the Claudia system, they can get very, very consistent dosing across long periods of time. So it's my prediction that before very long, this technology will be used in the clinic to improve efficacy and avoid toxicity in cancer patients. So for today, we need to be able to treat cancer patients. We need to develop new precision medicines. But as we look to the future, I think we can imagine a better time when we can avoid cancer altogether, when we can predict more accurately who's going to develop cancer and intercept it at a very early stage. And here I want to close with an example from Regina Barzilay, which does exactly that, using the power of artificial intelligence to predict the future of cancer. And this is a personal story for Regina because she herself developed breast cancer in 2014. And she, during that journey, asked her doctors, why couldn't you have known this was coming? You take an image of my breast every year. Surely there was something there at an earlier stage that would have told you that I was going to get cancer. And they sort of put their hands up and said, you know, we do the best we can. But Regina wasn't satisfied with that answer, and so she wanted to know perhaps there are latent signals in mammographic images that would tell you that there's a cancer coming at some future time. And so she then collaborated with colleagues from the Mass General Hospital and elsewhere to use AI technology and deep learning algorithms to ask the question, if you train computers with thousands of images from women who didn't get breast cancer in the future, or ones who did at 3 years or at 5 years, can you actually develop an algorithm that can accurately, or at least more accurately, assess the risk of an individual? And I'm happy to say that this has worked. In a paper published a few years ago, they demonstrated that they can develop a neural network which is quite predictive in assessing risk. It dramatically outperforms the standard used in the field today, which are based on things like family history of disease or age, but nothing about the particulars of the individual. The AI-driven model called Mirai greatly outperforms that technology. Because it was developed initially with a data set from a limited number of hospitals, they've now moved beyond and tested these systems with data from 7 different hospitals across 3 different continents. And the algorithm works equally well, if not a little bit better, with those diverse data sets, which is very, very encouraging. And this has now been implemented across the world. This slide says there's 1.7 million mammograms that have been screened by Mirai. I checked this morning, we're up to 1.9 million. So this is being dramatically rolled out to improve the doctor's ability to accurately predict and then monitor patients who are at the highest risk of developing breast cancer in the future. So I'll close there and remind you that we're celebrating 50 years of cancer research at MIT. It's been a long and remarkably successful journey, but I think the best is still yet to come. We at MIT have tremendous strengths across our great university to bring technologies from far and wide to apply to these difficult problems, and I think with the MIT HEALS initiative, we have potential to do that much more. So I'll stop there and thank you for your attention, and I'll now introduce my colleague Doug Laufenberger, who'll take it from here.
Good morning. I'm very appreciative to be able to participate here and provide a complementary partnership perspective from engineering, celebrating alongside the remarkable past of life sciences at MIT and anticipating the surely even more remarkable future ahead. So I need to start from this engineering perspective of what engineering contributes. Engineers create technologies to be useful to solve problems and address challenges in society. They do this by bringing together manipulation of components and making new components to construct these technologies. But they do it in a manner that's going to be predictive rather than trial and error, which involves quantitative measurement and modeling, so synthesis and analysis. And as was pointed out by our Dean of Science, you can't translate anything from nothing. And so the engineering technologies are grounded in particular branches of science, and that's really what defines the discipline of engineering, is what is its foundational science that is going to measure and model, and then it's going to manipulate and make from. So examples include mechanical engineering and electrical engineering, firmly grounded in the realms of physics, and chemical engineering, materials engineering, firmly grounded in the science of chemistry. Those are very well-established examples. And of course, what happens then is these engineering disciplines are aimed at a spectrum of application realms. Could be manufacturing, energy, transportation, environment, including medicine and health. And so engineers will bring together their analysis and synthesis of their branches of science to address these problems. And in fact, in the engineering world, ever since the 1950s, these classical engineering disciplines have been applied very effectively to medicine and health with outcomes such as prosthetics, kidney dialysis, heart pumps, imaging, drug delivery, and these all came from the physics and chemistry-based engineering disciplines that existed at that point in time. And so if you look back, you could call this really was mid to late 20th century convergence of engineering disciplines with medicine and health. Just that minimal biology was involved. And so when people talked about biomedical engineering, it was a big M for medical and a very, very tiny B for bio, because it really was not involved in a substantial way. Nonetheless, it was the right convergence for that time, pre the revolutions that Phil talked about. So since that time, since the 50s, 60s, 70s types of convergence, there have been indeed these two major life science revolutions, or waves. I like the notion of waves. Molecular biology being the first permits biological science to be fused with engineering, that engineers can use molecular and cellular components to make new biological technologies. So that enables a biological engineering. The second one being the genomic biology revolution, and that in fact requires bioscience to be fused with engineering on the highly quantitative measurement at scale modeling to understand and model in a predictive way these very complex molecular cellular systems, so that the new biological technologies synthesized from molecular biology revolution can now be analyzed in a predictive way based on the genomic biology revolution. So you see, those two revolutions laid the foundation for a brand new kind of engineering that was not possible before them. Not surprisingly, MIT is the place at which this new biological engineering discipline was created. In 1998, it was established, became a full-fledged department in 2005, and became Course 20. And what you see is that it now stands alongside the physics-based and the chemistry-based engineering disciplines, but now its science foundation is molecular biology and genomic biology. And from that foundation, it can make technologies in a predictively designed way to apply to manufacturing, energy, environment, defense, and medicine and health. So it's alongside those in being able to address the challenges across the entire spectrum of society. Now, I want to make sure I say something about education, because you really can't make long-standing waves or have a sustained revolution without educating the next generation of people who now think in the new ways and have capabilities in the new ways. So a very profoundly important thing about biological engineering here is it became Course 20, the first new MIT undergraduate major in 39 years, entirely new curriculum. Students were rooted in classes from the biology department in genetics and biochemistry and molecular biology, but all the engineering, analysis, synthesis, measurement, and modeling is brought to bear purely and solely on the molecular and genomic biology in an entirely new way that has never existed at any institution worldwide. And now these students are going on to become the next generation who are addressing these problems in ways that the physics-based engineering and chemistry-based engineering just don't have the capabilities to do. And so now we can add to the armament that the world needs. I will note at MIT, education is a huge priority. And in fact, I need to spend happily, unfortunately, most of my day in my office having office hours for my undergraduate students who have an exam tomorrow morning, and they want to discuss the questions and the problems and the topics. And to me, that's a huge priority, as it is for faculty across the Institute. So what are the products of this, along with new types of ideas and technologies? Of course, not surprisingly, companies have been spun out, and I loved Phil's list, and we could put biological engineering spawn companies alongside. I'm just going to note a few that are widely recognized as preeminent in their particular field. So Adimab out of Dane Wittrop's lab in biologics discovery and engineering, CN Bio out of Linda Griffith's lab in organ-on-chip technologies, Pivot Bio from Chris Voigt's lab in agricultural biotechnologies, Applied Biomath, now part of Certara in systems pharmacology from myself and a number of colleagues. Also note Strand Therapeutics at the bottom, now bringing synthetic biology into the realm of engineering immune cells to be more effective, again, in a predictively defined way, by Ron Weiss and Darrell Irvin. To end, in the last few minutes, in a forward-looking way, but of course to look forward, you say, where are we going? And of course, where you're going depends on where you're already rooted. And so one of the major things that's coming out of this discipline is addressing what I think is one of the long-standing challenges that has not been addressed effectively yet. President Kornbluth talked about the hardest problems. That's what this is. You all are very well aware of the attrition in the drug discovery and development pipeline, that a very, very small fraction of therapeutics that enter the clinic succeed. And the biggest problem of that is that the major costs of the pipeline are in the clinical trials, so you're wasting the vast majority of the money that you're investing. So this is a critical problem that's been around for decades. And again, people say, well, we're doing the best we can. We need to do better. Society needs it. So what is at the root of this problem? There's multiple issues, of course, but from our point of view, it's the very poor translatability from animal models to human patients, that one can do very, very, very effective things in animals that just don't work in humans. That's what's got to be improved. And why is that? Well, traditional animal-to-human translation focuses on using genomics largely and other quantitative measurements, identifying pathways and processes involved in a disease or lack of response to a therapeutic that are observed from very well-controlled animal experiments, and presume that if those are what's most important in the animals, then if we can find some hint of them in the humans, they're also going to be important in the humans. But this is incorrect. In the vast majority of cases, the things that are most important in the animals are not what's most important in the humans. Therefore, things that work very well in animals are not effective in humans. And this is the long-standing lament, again, you're all familiar with, of curing disease after disease in mice, with no consequent benefit in humans. I call this the tip of the iceberg problem, because what you're looking at is what's most important in the animal with very little guarantee, and in fact, very minor chance that it's actually what's also important, most important for the human patients. How do you address that? Well, this is where the combination of biology and engineering inherent in this new discipline really holds a lot of promise. That if you understand on the one hand what these biological pathways and processes are, what the data represent, and on the other hand, you're capable of the sophisticated computational analysis and modeling one can bring together, for instance, in the example I'm showing here, using a semi-supervised AI machine learning approach, in which the secret sauce is not the sophistication of the computation, but it's the understanding of how the data map and what the data mean biologically. And you can find pathways and processes that are, in fact, most important for humans, even when they seem to be of negligible importance in the animals. So I call this, instead of being focused on the tip, you can actually find the iceberg underneath that's most important for the human by this combination of omics data, understanding biological pathways and processes, and the computational machine learning. These are projects we've been pursuing, not only with a number of the biopharma companies in this area and elsewhere, but the Department of Defense, the Gates Foundation for Vaccines, and not surprisingly, the FDA, which wants to improve this and the methodology for this strongly. Some examples have to do with Humira resistance and Crohn's and colitis with a very well-known biopharma company, with the Army Protection Against Neurocognitive Trauma, and I can name others. And most importantly, this approach is now being adopted into industry therapeutics pipelines, and so I believe that we will see over the coming decade or so significantly improved translatability from the preclinical studies to clinical. So that's just an example of rooted where we are now in biological engineering, looking at the kind of challenge that has been too hard to do before, but is no longer too hard. With that, I thank you for your attention. I thank you for being here at this momentous event. And it's a great pleasure to introduce our next speaker, Ruth Lehman, who's a professor in biology and director of the Whitehead Institute.
Hello, good morning. It's a real pleasure for me to be here today and to celebrate HEALS. Like Sally, I am also a recent recruit, and I also have to say that it is just invigorating and empowering to be in an environment where the university is without limits, and the environment, the ecosystem of Kendall Square could not be better, and the hospitals across the river give us really an opportunity to think big. What I want to tell you about is a little bit about the Whitehead Institute. And I'm calling it a radical experiment. This is actually a title of a book about the history of the Whitehead Institute that Jerry Fink just completed. And at the beginning, when the Whitehead Institute was founded, it was clearly a radical experiment, and it wasn't always taken with total positivity, let's say, because it was a bold approach, having an institute which was independent in its research, government, and finance, solely associated with an institution, with MIT, where all the faculty are teaching at MIT and are in departments at MIT. The founding vision was very bold as MIT is, to assemble the best scientific talent, to catalyze their work with support, and to encourage them to go their own ways. There were many early breakthroughs, and actually you heard about it quite a few. You heard several times about the breakthrough work of Bob Weinberg, and that was at the Whitehead Institute in the field of cancer, work in stem cell biology, transcription, and other fields. In particular, and again, you heard this already, 1/3 of the human genome was sequenced at the Whitehead Institute in collaboration with the NIH and MIT, and that led then by Eric Lander starting the Broad Institute as a spin-out. What's also really important is shaping a new generation, not just as teachers, but also the Whitehead Institute established the Fellows Program for newly minted PhDs. And the first notable fellows are David Page and Eric Lander, and others are George Daly, Angelika Ammann, and Peter Kim, and also David Bartel, who was mentioned earlier. So what are we about today? Today we are 18 members, 14 of them are tenured, and we have 6 fellows, and we have a large number of graduate students and undergraduates and postdocs working in our laboratories. And probably one would say from just the recognition, the experiment was a success, but it's still an experiment as I see it. So what are we looking for? And when I speak here about the Whitehead, I think I speak about biology, I speak about basic sciences. We really today want to understand how organisms perform their tasks in life, and that is to capture the complexity. We believe that that has to be achieved by a mechanistic understanding, as we heard Phil Sharp saying, measuring. We want to understand it mechanistically, but then we do want to deconstruct and reconstruct to really understand complexity of life. And that can be done only by collaborative work, and that is to draw from biology, physics, chemistry, engineering, and increasingly computer science, AI, and machine learning to develop new tools and designs. In general, we don't want to be afraid of asking any questions. We don't want to be afraid to change directions if that is important. And I think HEALS will empower that through the collaborations that will spark new ideas and new points of inflection. So 2 points when we think about history, we can jump at today, we can analyze cells now at a level of resolution, and we can ask and model cells in a way that was unforeseen before, leading to a project spearheaded by Jonathan Weissman and also with the work of Ian Cheeseman to describe actually a virtual cell, to know every component, every aspect of what is happening in a cell. But going one step further, how can we understand a whole organism? How do we understand the parts? How do we bring neurobiology and immunology together to understand how an organism actually functions, how it feels an infection? So these projects are supported by some initiatives, and these initiatives, they're trying to be very bold, and they're trying to really address some of the pressing issues of today's challenges. So the Whitehead Initiative of Biology, Health, and Climate Change addresses questions directly of agricultural questions in terms of drought-resistant seeds, but also how does temperature affect the cell? How does a cell work at different temperatures? And you will hear later in the next session from Sinisha Harvartan about hibernation. The Women's Health Initiative brings fundamental science together with really understanding why is there a health bias, and I'll talk to you a little bit later about this. And all of this could not be possible grasping this complexity without using AI approaches to lead the way, but also to be led by the data that we are accumulating. We are not alone. I always see the Whitehead a little bit as a hub with have lots and lots of spokes. Many of those spokes go across the street to MIT, but also we have lots of interactions with hospitals and other universities. We seek out collaborations, and collaborators seek us out. So today, what I want to talk to you is actually give you 5 quick vignettes about some science, and they're pretty much all going on right now. these vignettes are also highlighting how we're perhaps addressing somehow Doug's criticism to basic science that we're doing this all in model organisms, because as you can see, much of what we're learning here is directly either applied to humans, in humans, or not in humans, but in plants. And so I will be talking about these 5 challenges in biology and actually for human health and the planet. And so let's jump into it. First, I'm going to talk to you about the Loreto Lab. The Loreto Lab works on toxoplasma, and many of you know how dangerous toxoplasma can be, as it infects a large percentage of the population and can have really serious consequences for pregnant women, infants, and immunocompromised people. So what Sebastian and his group have done is they took all the 8,000 genes of toxoplasma and made gRNAs against them so that he can interfere with those RNAs and then infect the organism where normally the infection occurs, and then they're able to use different tissues and look at what of these cells are now surviving. Those cells which are surviving are obviously not the ones whose genes are required for the function. And so he can directly select with this whole genome mutagenesis for genes which affect fitness, and has identified a number of genes which affect the fitness of the infection. And this is clear how it's directly applicable in the future. The next, we go to another challenging problem, and that is how do we make better drought-resistant plants? How can we deal with the issue of food supply? And so what most of you know is that many seeds are actually produced by crossing different parent plants, and then the seed itself is much better than the parents were. The problem is that those seeds cannot be propagated continuously, and so if they're just grown, then there will be a large variety. So what Mary Gehring is addressing is to ask the question of, could we actually make seeds which can be asexually propagated? And that actually happens sometimes naturally. And so she's using these kinds of seeds, which sometimes make that mistake of not needing fertilization, to find out what factors are required, and then can we up the opportunity for seeds to develop asexually and then produce populations of seeds that can actually be used without having the hybrid vigor problem. The next question is going directly to human biology, and that is addressed by David Page. In his lab, really is puzzled by this striking and still very unexplained sex bias in the prevalence, the severity of diseases between males and females, also certainly drug sensitivity. And so what they found is really surprising and very interesting. So when we just look at all the cells in our soma, in our body, not the cells in our reproductive organs, there are 2 types of chromosomes, sex chromosomes, the X and the Y. And when we look at the expression of the active X chromosome in males or females, so XX versus XY people, the active X is expressed the same way. So all the genes, there's no sex difference. But the sex differences come from the genes which are expressed on the so-called inactive X, which is actually not inactive, but expresses 38% of the cells, of the genes on the active X. And also there are genes expressed on the Y chromosome, and these could really influence the expression of these genes are making a cell different, any cell in the body different, whether it is in an XX organism or an XY organism. And so the idea is that this can have an influence on disease prevalence and sex bias. The next issue is another big question, and that's the big question of neurodegeneration, and there is so little that we know at this point where we can really do something about neurodegeneration. A particular type of neurodegeneration is neurodegeneration caused by prion disease. Prion disease is usually a very swift neurodegeneration, and the death is due to the accumulation of misshapen proteins. And so Jonathan Weissman designed CHARM, which is where a fusion protein recruits a methyltransferase specifically through a DNA binding domain to a specific gene. And by that, it methylates that gene and silences that gene. And so this can be delivered into the brain, and in mice, this reduces 80% of the prion gene expression, and mice which are affected by prion disease live longer after this injection. And so my final vignette is perhaps the biggest challenge in human health, and that is chronic disease. There are many types of chronic diseases, and the problem with chronic diseases is they're not due to a single gene, so we cannot start with a single gene. So, but they have multifactorial causes, and we don't even exactly know what the causes are. So what Rick Young and his colleagues found is that often through elevated oxidative environment, through the pathogenic factors, proteins are less mobile. They are less mobile because their cysteines are altered so that they are now interacting with other proteins more and putting through cysteine bridges. And he phrases this as proteolethargy, and this proteolethargy may, first of all, lead to reduced function and output, and they showed this very clearly with insulin receptor mobility being reduced, and it is also a target which can actually be more globally and widely used. So I hope that with these vignettes that I just presented, you got an idea of where basic science meets translational science, and that couldn't be more better illustrated than the many companies which have sprung out of these initiatives that have started at the Whitehead Institute. I'm really looking forward for HEALS to kind of further enlarge the interactions that we have, and I think the possibilities for science, both at the mechanistic level and understanding the complexity for health and the planet, is really, really great, and I look forward to that. Thank you very much.